Hello, greetings, and welcome to another of my online lectures. I am Professor Jun Cajigal. So for this video, we shall be tackling the use of the microscope. So before we move on to our agenda for this video, don't forget to like and share this video so that we could reach more people, especially the students who are taking biological sciences. And also don't forget to subscribe to my YouTube channel, The Science Guy Professor Jun Cajigal. Don't forget to subscribe and click on that notification bell so that you would be updated on our future videos. Alright, so let us now go into the details on the use of this microscope. Alright, so in my first video, I mentioned that the microscope is an instrument that produces a magnified image of very small or minute objects. And this microscope is a compound microscope. A compound microscope has two major lenses. We have the objective lens and the eyepiece lens or the ocular lens. The objective lens collects the light from the specimen and produces an initial image. Then that initial image is further magnified by the eyepiece lens or the ocular. Alright, now before using the microscope, be sure that you can carry the microscope properly. Now the proper way of carrying the microscope is that one hand should support the arm, this is the arm of the microscope, and the other arm should support the base. So one hand should support the arm and the other hand should support the base of the microscope in transporting the equipment from one place to another. Alright, then after you have placed the microscope here on our working table, then you can plug your microscope. So this microscope is equipped with an electric light source, unlike other microscopes in which the light is collected through a mirror. This microscope has already a built-in electric light source. So you can just plug the microscope and you're ready to go. You have here a switch at the base of the microscope that you can turn on, so you can turn on the light bulb. Alright. Alright, let's talk about now the preparation of the specimen for microscopic observation. Now, usually the specimen is mounted on a glass slide, and the specimen is covered with a small piece of glass called a cover slip. So we will be using a simple technique on preparing the specimen. This is called wet mounting. So in wet mounting, you place the specimen at the center of the glass slide and add a drop of water. And then you cover the specimen with your covered glass. Then you place the specimen onto the stage of the microscope. Now, using the coarse adjustment knob, you raise the stage up to its maximum. Alright? Then, you look through the ocular lens and gently lower the stage using the coarse adjustment knob until you can see the image of the specimen. So that's how you focus the specimen, by lowering gently the stage of your microscope. Okay, using the coarse adjustment knob, but be sure that your objective lens is in low power objective. Alright? Alright, so that's how we focus the image using the coarse adjustment knob. And using the low power objective. Now, if you want to increase the magnification of your image, then you shift your objective lens from low power to high power using the revolving nose piece, and look through the eyepiece and just turn the fine adjustment knob. The fine adjustment knob is for final focusing of the image so that the image of your specimen would be clear and crisp. Alright, so use the fine adjustment knob for final focusing so that your image would be crisp and clear. And you use this only for the high power objective lens. Now, we can adjust or improve the image produced by the microscope simply by adjusting the contrast. So it's like adjusting the contrast in your television set. If you want to improve the quality of the image produced by your TV, you simply adjust the brightness and including the contrast so that you will have maximum viewing pleasure. It's the same thing with the image produced by the microscope. We can adjust the contrast of the image so that we can observe more details of the image. Now, how do we improve the contrast? There are two ways of adjusting the contrast of the image produced by the microscope. We can use the diaphragm. So the diaphragm can be opened or closed in order to regulate the amount of incoming light. And in that manner, we can improve the contrast. Now, there is a lever here. So if you turn the lever like this, you can open and close the diaphragm. In this case, we can improve the contrast of the microscope. Another way of adjusting the contrast is by dimming the light source. So at the base, there is a dimmer knob. You can turn the dimmer knob to adjust the illumination of your light bulb. Alright? So those are the two ways in which we can adjust the contrast so that we can improve the quality of the image produced by the microscope. Now, there's another way of improving the contrast of the image produced by the microscope, and that is through staining. Now, staining involves the use of certain chemicals, such as iodine or methylene blue. So, you simply get the glass slide and you place the specimen at the center as usual, just like in wet mounting. Then you just add iodine onto the specimen, then cover it with the cover slip. Now, you may want to use methylene blue instead of iodine. It's up to the one who will observe or use the microscope. So, you get the glass slide, place the specimen at the center of the glass slide, then add a drop of iodine or methylene blue, then cover it with the cover glass, and you are good to go in observing the specimen under the microscope. So with that, you can increase the contrast, and by the time the nucleus would be visible under the microscope. Now, in using the microscope, we need to consider these two important aspects. Number one is magnification, the other is resolution. Now, what do we mean by magnification? Magnification simply means the ability of the microscope to produce an enlarged image of the specimen. And how do we determine the magnification of the microscope? Well, we simply multiply the magnification of the objective lens with the magnification of the ocular lens or the eyepiece. Now, for example, let's say the magnification of the objective lens, let's say low power objective is 10x, then the ocular lens has a magnification of 10x. So the total magnification of your microscope is 10 times 10. The microscope enlarges the image of the specimen 100 times. Okay, so you multiply the magnification of the objective lens with the magnification of the eyepiece or ocular. Now, what about resolution? Now, resolution simply means the ability of the microscope to distinguish two very close points. Or in short, how clear or how crisp the image is so that you can distinguish between two very close points. And that's what we meant by resolution. Now, as we increase the magnification from low power to high power, usually the resolution of the microscope decreases. As we increase the magnification of the microscope, the resolution decreases. Increase magnification, you decrease the resolution. So, in order to improve the resolution, you either improve on the contrast, you adjust the diaphragm, or you adjust the dimmer knob so that you can achieve the best image for the microscope. Now, one final note before we end this video. So, be sure that you clean your glass slide and cover glass or cover slip. Alright? Then, be sure that your objective lens is set to low power. So, turn your revolving nose piece and be sure that the objective lens is in low power setting. Okay, turn off your light source like so. Unplug the microscope. Okay, be sure that your cable is organized like this. Just coil it like so. Alright, and you are now ready to return the microscope to our lab technician. So be sure that one hand supports the arm and the other hand supports the base of the microscope. Alright? Okay, so I guess that's about it for this video on the use of the microscope. So please like and share this video so that we could reach more people, especially the students out there who are taking biological sciences. Don't forget also to subscribe on my YouTube channel, The Science Guy, Professor Jun Cajigal. Don't forget to subscribe and click on that notification bell so that you would be updated on our future videos. So I guess that's about it. In our next video, we shall study the different types of microscopes. Alright, so live long and prosper, and always remember to always keep your space and always love science. So long for now. See you on our next video. Bye-bye!